and I will present you our study done with bestatranla titled Identification of Competing Endogenous RNA Regulatory Network to Explore
Again, it doesn't show the video.
Fibrosis. Idiopathic
I have available this video if you want to do it.
pulmonary fibrosis. Hi, I am Muhammed Fatih Kırcalı, and I will present you our study done with bestatranla titled Identification of Competing Endogenous RNA Regulatory Network to Explore Pathogenesis in Idiopathic Pulmonary Fibrosis. Idiopathic pulmonary fibrosis, in short IPF, is a chronic progressive interstitial disease with poor prognosis. As name suggests, reasons behind IPF is still unclear. Therefore, we wanted to investigate molecular mechanisms of pathogenesis using systems biology tools. Datasets from Gene Expression Omnibus are used for mRNA and macroRNA expression assessment, and differentially expressed genes are identified. Then interactions between molecules are obtained from databases BioGrid, MirTerbase, MirDB, and CertBank. All interactions are plugged into cytoscape, protein-protein, microRNA, mRNA, and finally circular RNA, microRNA, mRNA interactions network, which constitutes our competing endogenous RNA network, are all constructed. Hub nodes and modules are determined using CytoHubba and M-Code plugin apps. Differentially expressed genes are also enriched using Metascape Express Analysis tool. 3 RNA sequencing datasets for mRNAs yielded 724 differentially expressed genes. 3 microarray datasets for microRNAs are analyzed, and totally 278 differentially expressed microRNAs are obtained. Enrichment analysis results yielded nabamatrosome-associated cilium movement, humoral immune response, and extracellular matrix organization as most prominent summary terms. All these terms are implicated in IPF pathogenesis in previous literature. Next, protein-protein interactions network is analyzed to determine hub proteins. A hub protein, estrogen receptor 2, is considered to have a protective role against inflammatory diseases. Here we think it may have a possible role to explain disease progression difference between male and female patients, in which worse prognosis is in male patients. Hub proteins TP63 and TP73 are also interesting, as they are commonly having a role in regulation of development, apoptosis, cell senescence, and tumor suppression. In IPF, TP63 also has a role in mesenchymal type aberrant basal cell expansion. Role of TP73 in IPF is not clear, but it is implicated in driving differentiation of multicellular cells in lung epithelium. Therefore, we consider TP73 may have a role in upregulation of ciliary elements. From competing endogenous RNA regulatory network, 2 modules are identified. First module contains serine protease TMPRSS4, encoderin, and macroRNA 543. All members of this module are implicated to have a role in regulating epithelial mesenchymal transition, which drives loss of proper epithelial healing and constant expansion of fibrotic cells. Lastly, second module includes adrenergic receptors and macroRNA 484. Adrenergic receptors are found to be downregulated in other studies similar to our results. But recent research also proposes antagonism of adrenergic receptors to have a therapeutic effect. Therefore, the role of adrenergic receptors in IPF is still unclear and more research is needed. So in this study, we identified molecular signatures of IPF among differentially expressed mRNA, microRNA, and computationally predicted circular RNAs. We hope these results may help to uncover molecular mechanisms behind IPF in the future. Thank you.
Thank you, Mohammed. Beautiful presentation. I want to make a question. Do you validate your results? I mean, did you find biological evidence after this bioinformatic analysis you did?
Currently, actually, we are continuing our analysis with our module genes because we need more insight about them to continue with the experimental works. Only after experimental works we can claim a biological exact relevance to the disease.